Recombinant DNA technology. A complete overview definition. Recombinant DNA technology involves joining together DNA molecules from different organisms and inserting them into a host organism to produce new genetic combinations for research, medicine, agriculture, and industry. Basic principle. The central idea is to cut a desired gene using restriction enzymes, insert it into a suitable vector, and introduce it into a host organism to express the gene or replicate it. Tools of recombinant DNA technology. Enzymes. Restriction endonucleases, molecular scissors that cut DNA at specific sequences, for example, ECO-RE, HIND3. DNA ligase, joins DNA fragments by forming phosphodiester bonds. Polymerases, Taq polymerase, used in PCR to amplify DNA. Reverse transcriptase, converts mRNA into cDNA. Alkaline phosphatase, removes phosphate groups to prevent vector self-ligation. Vectors carry the foreign DNA into the host. Plasmids, most common bacteriophages. Cosmids, BACs, bacterial artificial chromosomes. YACs, yeast artificial chromosomes. Ideal vector features, origin of replication, or a selectable marker gene, for example, antibiotic resistance. Cloning sites, MCS. Host organisms. Bacteria, E. coli, most commonly used yeasts, Saccharomyces cerevisiae. Plant cells, Agrobacterium tumefaciens. Animal cells. Steps in recombinant DNA technology. Isolation of genetic material. Extraction of DNA using physical, centrifugation, and chemical methods, lysis buffers, phenol chloroform. Cutting of DNA at specific sites. Use of restriction enzymes to cut DNA into fragments. Produces blunt or sticky ends. Amplification of gene of interest, optional PCR, used to amplify the gene segment for cloning. Ligation of gene into vector DNA. Ligase enzyme joins foreign DNA with vector DNA. Insertion into host, transformation techniques, heat shock, bacteria electroporation. Microinjection, animal cells. Gene gun/biolistics, plants. Agrobacterium mediated transformation, plant selection and screening. Use of antibiotic resistance or reporter genes, for example, GFP, LACE. Blue-white screening for successful recombinants. Expression of recombinant gene. Transcription and translation of inserted gene in host. Production of desired protein, for example, insulin, enzymes. Downstream processing. Isolation, purification, and characterization of the expressed product. Applications of recombinant DNA technology. Agriculture. Bt cotton, pest-resistant golden rice, vitamin A-rich herbicide-resistant crops. Medicine. Production of insulin, growth hormones, vaccines. Gene therapy, for example, SID treatment. Diagnostic tools, for example, ELISA, PCR tests. Research. Gene function studies, for example, knockout/knock-in. Protein engineering. Functional genomics. Industrial enzyme production, for example, amylase, protease. Bioplastics and biofuels. Ethical, social, and legal issues. GMO safety concerns in food and environment. Patenting of life forms and biotechnologies. Gene editing ethics, CRISPR/Cas9 in humans. Risk of bioterrorism. Requires strict regulation and public awareness. Examples of recombinant DNA products. Product organism application. Humulin, insulin, E. coli. Diabetes treatment. Hepatitis B vaccine. Yeast immunization. Flavor saver. Tomato delayed ripening. Bt cotton pest resistance in crops. Erythropoietin. Mammalian cells treat anemia. Advanced concepts. 1. Expression vectors. Engineered vectors that ensure high-level expression of foreign genes. Include strong promoters, for example, CMV, T7, enhancers, and terminators. 2. Reporter genes used to identify and track gene expression. Examples: GFP, green fluorescent protein, LACE, luciferase. 3. Gene silencing and editing RNA interference, RNAi, to silence specific genes. CRISPR-Cas9 for precise genome editing. 4. Synthetic biology designing and constructing novel genetic circuits and pathways. Summary chart step process tools involved: 1 DNA isolation buffer, enzymes, 2 gene cutting restriction enzymes, 3 amplification PCR, 4 ligation DNA ligase, 5 transformation heat shock, electroporation, 6 selection marker genes, 7 expression host machinery, 8 purification chromatography, centrifugation.